I'm going to introduce this next topic and get into it a little bit more in the synchronous lecture. I just wanted to get us a little ready for that by talking about some basic principles in antibody-drug conjugate chemistry. It's a pretty cool idea, and it's nice that it interfaces very closely with biology, and it's really where a lot of modern drug discovery is heading, so it's good to learn a little bit about it. Alright, so what are the things we're going to talk about? I'm going to first introduce some examples of commercial antibody-drug conjugates to kind of see the key parts of the structure, the organic, chemical, relevant stuff, and maybe where the antibody is, linking methods, etc. We're going to talk a little bit about how antimitotic drugs work, and because a lot of these drugs are antimitotic and anti-cancer drugs, how do these drugs work biologically? Next, we're going to get into the synthesis of tubulysin, which is one of the molecules that is used in a lot of these antibody-drug conjugates. And then we're going to talk about development of tubulysin analogs as a new class of ADCs, antibody-drug conjugates. So mostly our goal is to get through these two papers. I might do this one today. We'll see. Otherwise, I'll do it all next time. And it just, when it's synchronous, I can answer questions a little bit easier. So this is not that hard, though. Well, maybe I'll do a little bit of it today, a little bit next time. We'll see. Okay. Alright, so let's show some simple examples of an antibody-drug conjugate. And I guess I'm using the word simple sort of loosely because they're not very simple. I mean, but I guess the basic idea of these can be shown pretty easily, and what the key organic chemistry is that links maybe a drug to an antibody. Okay, so our first example is a drug called brentuximab vedotin, or Adcetris. A lot of these are anti-cancer drugs, and I think there's additional antibody-drug conjugates that treat other diseases. We're going to focus on the anti-cancer ones. So there's a couple components. There's an antibody component, which is a protein that recognizes other proteins on the surface of the cell, particularly like an anti-cancer or like a cancer cell. It's called brentuximab. MAB is the antibody. It's this 3 letters that represents a huge macromolecule. There's some kind of attachment point where the antibody gets attached to the drug conjugate. A lot of these antibody-drug conjugates have a cleavable linker, so some kind of cleavage mechanism. Now, often they use a protease, like cathepsin. Cathepsin is a protease that cleaves, I forgot the exact site specificity, but I think it will cleave next to the citrulline, which is a basic amino acid, and it's kind of related to lysine. So it recognizes specific amino acid sequences, and it's it cleaves the linker. Okay. Then there's a spacer group, and then there's some kind of warhead. This is a typical arrangement of an antibody-drug conjugate. The warhead does the anti-cancer activity, and like as we'll see, these prevent mitotic spindle assembly, so it basically inhibits mitosis, and in a cancer drug that works really well to basically kill cancer cells. So the linker component is valcit, a dipeptide. It's cleaved by the protease cathepsin. And the spacer unit is 4-aminobenzyl oxycarbamate, which is this kind of thing right here. It separates the linker from the warhead. So carbamate is this functional group. It looks like kind of like an amide on one side, almost like an ester on the other side. You put them together and you have a carbamate. And then the warhead in this case is monomethyl auristatin, or MMAE. We'll see that one in a few more places. It's a cytotoxic peptide that binds to tubulin and basically inhibits mitosis. So that's overall what this antibody drug conjugate is. In 2011, it's approved by the FDA to treat Hodgkin lymphoma. It's basically cancer related to white blood cell lymphocytes. Also, a different type of lymphoma due to kind of T lymphocytes that are acting irregularly that causes cancer. And then in 2017, another type of lymphoma, cutaneous, is like related to the skin, and so it's a pretty well established antibody drug conjugate used to treat cancer. Annual sales $600 million. I mean, this is expected to peak at around $2.7 billion. So these drugs make a lot of money, and that's why a lot of drug companies are interested in them. As a kind of a novel way to treat disease, often with fewer side effects. Okay, here's some more examples. Here's a cool paper. Actually, it's a book, and I think I have a copy of this somewhere. Predictive markers in oncology. So this shows a couple more examples. This one is called gemtuzumab ozagemicin. They have fun names. This is what we call a first generation earlier antibody drug conjugate. So it's got the antibodies, this crazy thing up top. It's a protein. And it has a linker. This is a cleavable hydrazone linker, so that I think under aqueous conditions, the hydrazone gets cleaved. Not immediately. I mean, it has to be somewhat slow, otherwise it'll just degrade immediately. And it's connected to this crazy thing called caliciumycin. Caliciumycin is a very interesting molecule, and it's a natural product, I think produced by bacteria. It's got this crazy thing called a enediene right there, enediene, so two alkynes and an alkene. This thing cyclizes to make kind of like an aromatic diradical. It's like a benzene with two radicals on it, and that can that's like super cytotoxic. It's also used as an antibacterial, it has antibacterial activity. I don't think it's used as a drug because it's a pretty complicated molecule. What else about this? Well, you could do a Wikipedia search on it if you're interested. Caliciumycin, and it's a very fascinating substance just by this structure. And that cyclization mechanism is called it's called a Bergman cyclization, which is how the enediene cyclizes to form an aromatic diradical, and that's just super. The radicals will start ripping off atoms within a cell and destroy it. Basically, it's very cytotoxic. Random license. What do I mean by that? So one of the reasons it's first generation is that the surface of the antibody has some lysines, nucleophilic lysine groups, and it says 2 to 3 caliciumycin warheads per antibody immunoglobulin. And what does that mean? Why is it 2? Why is it 3? It's so there's random lysines, and this what they do is they take this whole thing and they put a leaving group on it, like a succinimide or something, right there, and then expose it to the antibody, and it just gets attached at random locations. That seems a little weird. Usually a drug is a very specific thing with a very specific structure, but this may be attached at a couple different places. So that's one of the kind of earlier methods to make antibody drug conjugates. We have more modern methods that are a little more homogeneous and consistent. But this is one example of one of these. This is a second generation antibody drug conjugate. This one uses a non-cleavable thioether linker, so it's attached also to lysines. So there's I think these little hexagons show kind of the attachment points to where the drug gets attached, a little bit randomly, and it's a little inhomogeneous, and it's non-cleavable. That's weird. Let's talk about that in a second, in a minute or two. How could it even work if it's non-cleavable? Like, would this get into your cell and then stay non-cleaved, and then have this go inhibit mitotic spin assembly? That's kind of weird. How would it even work if it's non-cleavable? This at least is cleavable with a hydrazone. This is non-cleavable. But anyway, we'll talk about that in a second. Yeah, this is called, I think, Cadcyla, I think it's called. Okay, and then we have two more at the bottom here. The first one is over here on the left. This is what they're also calling a second generation antibody drug conjugate. This one has a protease cleavable linker, so this is probably cathepsin again. It's a couple carbons and a nitrogen. I don't think that's based on lysine, because I think lysine has 4 carbons. I think that's right. So it's a kind of a weird amino acid, non-natural amino acid. And then they have that thing we were just talking about, monomethyl auristatin, I think it's called. And that is the warhead. This does the anticancer activity. This is connected to cysteines, and so the connection is kind of cool. The way it works is cysteine, which is SH, reacts with this thing, this whole thing. But imagine it with with an alkene there. With an alkene, that is kind of like a Michael reaction or organic reaction, where the SH attacks the alkene, and that's because these two carbonyls. That's like one of the kind of a modern linkage mechanism for antibody drug conjugates. Take the cysteine, and they often actually engineer cysteines, like they put them in certain places of the antibody with molecular biology, and then these things attach very specifically. So here, because of that, they actually get 4 of them attached per antibody drug conjugate, so it's a little more homogeneous and consistent. Okay, and then here's a third generation one. It's a kind of a little more complicated. They also use a non-cleavable linker. A non-cleavable linker. They use that same the cysteine technology to attach it to a maleimide, which is this thing. Imagine this, but with a double bond there. And so it's like a Michael reaction. The sulfur attacks. It's a nucleophile electrophile thing, and it snaps on. And they get very specific attachment, and they get like 4 of them per antibody drug conjugate. And let's see, they have a non-cleavable linker, so we'll talk about that again in a second. Weird that you can do a non-cleavable linker. And then here's the warhead. I think this is tubulysin. This is a different peptide, tubulysin versus MMAE, monomethyl auristatin. This one works a little bit better. That's part of why it's a third generation molecule. Okay. Oh yeah, this FC binding, we'll talk about that in a second. They mutated a specific lysine to a phenylalanine. I think to prevent more of the conjugate to be attached. They only wanted it on the cysteines, right? So there's a couple cysteine linkages, but they don't want to attach this to lysines, so they mutated lysine to phenylalanine. That'll do it, right? Okay. So So these are commercially available antibody drug conjugates. How would a non-cleavable linker even work? We just ask that question. How might it work? If it's non-cleavable, like this third generation molecule would go into the cell, would it just stay attached forever? And would this actually sneak into the nucleus and still allow this to work, even if it's a non-cleavable linker? So it's interesting because with non-cleavable linkers, the idea is the antibody gets internalized into the cell, so the protein does get sucked into the cell, but then it gets digested by the thing called a lysosome. So it sees this foreign protein and it destroys it. So it kind of does cleave, but not through like cathepsin, like a protease. So this gets kind of digested up, and this does ultimately do its anti-cancer thing. So that's kind of how an antibody drug conjugate would work without a cleavable mechanism like a cathepsin cleavage site. All right. Okay, so another cartoon diagram of how these basically do their job. This is from Current Strategies and Applications for Precision Drug Design and the journal Frontiers of Pharmacology. So this is an example of an antibody drug conjugate. You have the antibody, you have a linker, you have a payload, and the antibody likes to attach to some kind of protein or receptor on the cell surface. And there's actually two regions of the antibody that I should mention in terms of antibody naming or structure. The Fab region and the FC region. The Fab region is sort of the top region, and it recognizes the antigen, which is the thing it binds to. So it's very specific and binds specifically to that protein. And ideally it's a protein related to like a disease, like cancer or something. The FC region is the bottom, which binds to other receptors on the cell, but it's not really the antigen usually. So the FC region, this bottom part, is not really involved in the detection of antigens. And that's why you see when you see this, and you see it kind of flip over. And that's what this is. It actually binds. Okay, it binds, and then the idea is that the bound complex internalizes into the cell, and then sort of falls apart, and the cell surface protein gets recycled and kind of exported out to the cell again, and it kind of repeats the process. But then the antibody drug conjugate somehow gets degraded, maybe cleaved, maybe cathepsin protease helps out. And then the drug is eventually released into, it has to be imported into the nucleus, and then it kind of either binds to DNA or it interacts with microtubules and kind of inhibits mitosis. That's what we're going to see in a second for those peptides we were just talking about. Alternatively, you could do this with a ligand, targeted drug, some kind of ligand that would bind to a protein and then kind of get internalized, and same kind of thing. But antibodies do this really well. So that's why, and it's pretty easy to create antibodies now to target cells. A little harder to create a specific ligand that binds to a protein. But that's another strategy. Okay, so how do antimitotic drugs work? And let's review the basic concept of mitosis. All right, so let's talk about the cell cycle first, in very brief terms. You probably learned a lot about this in biology classes. So the cell cycle just kind of operates continuously between different phases, interphase, different stages of interphase, the S phase, the G2 phase. Mitotic phase is where all the kind of interesting stuff happens related to mitosis and cell division. and cytokinesis, which is the separation of, like, two cells break apart, two daughter cells, and then after those cells break apart, it returns back to another interphase, and the interphase is usually where cell growth and DNA synthesis occurs. This kind of shows what these different phases are. Interphase, at least in the DNA synthesis part of this, basically starts synthesizing two identical copies of the DNA inside the cell, and then you have two different copies of the chromosomes, and they kind of in this prophase part, the chromosomes kind of they spiralize in a way that they can be sort of separated. It becomes ready to separate into two different cells, I guess. And in metaphase, we form this thing called a mitotic spindle that uses microtubules to actually just start separating the two forms of the chromosomes, the two copies of the chromosomes. So it forms this mitotic spindle thing, and then using the microtubules, the chromosomes get separated into this anaphase part of this. Okay, so it's like we'll show a structure of how this actually looks in a second. But basically, the chromosomes get kind of attached to the microtubules, and then the microtubules sort of pull them apart, and now you have the separated daughter chromosomes, right? Because you're going to separate these into two cells here in a second. So then in telophase, now you start forming two different nuclei. So the nuclei get separated, and then cytokinesis occurs, and you have two new cells. So that's kind of how cells divide, right? Cancer, in a sentence, is a condition of abnormal cell growth caused by various factors, environmental and genetic. Of course, cancer is a huge thing, and we could, you could take a whole course on cancer. But let's just leave it at that for now, and say that one anti-cancer approach is to disrupt the construction of the mitotic spindle. So if you can kind of prevent this spindle from forming, if you have abnormal cell growth, you can kind of block cancer and tumor growth. And drugs that do this bind to inhibit the assembly of microtubules. They can also there's multiple ways that the molecules can interact with microtubules that we'll see, but that's like the microtubules are these little black lines, and by doing that, it kind of just it inhibits this separation of the chromosomes. Okay, so this is what it looks like, mitotic spindle during metaphase and during mitosis. So these green things are the microtubules, the blue things are the chromosomes, the little red dots are called kinetochores, I think they're called. Microtubules, green. Kinetochores, they're little pink attachment points for where the chromosomes and the attach to the mitotic spindle, okay? So what kind of drugs do this? Well, there's a lot of them, a lot of anti-cancer drugs, and primarily as an organic chemist, I love organic chemical structure, so I love seeing molecules like this. Vinblastine, it's an interesting organic molecule. You have these kind of indole things. There's this sort of cross-link in the middle, a lot of chirality, and interesting molecule for synthesis and everything. Vincristine is another very complicated molecule with also these kind of indoles and very complicated structure, a lot of chirality, dense chiral center. So a lot of the early organic synthesis accomplishments were molecules like this. Vinflunine is another one that also has kind of a dense collection of chirality and a lot of interesting rings and macrocycles. I think this one with the difluoride is probably man-made, maybe to enhance bioavailability or something like that. I don't think that nature produces difluorides, but humans do, and we often do that for to enhance pharmacokinetics and stuff like that. Taxol is a very famous one that also binds to microtubules, and it's used as a clinical anti-cancer drug. My postdoc advisor when I was at UCSF, Kiplin guy, he was the first one to actually synthesize this with Casey Nicolau. Casey Nicolau is like a world famous organic synthetic chemist at Scripps Institute. So my old postdoc advisor, that was his big accomplishment. And it was synthetically, that's a very challenging molecule to make. Epithylones are different, but they also have the same kind of general activity. Macrocycle, it's a cyclic ester, a macrocyclic ester, a macrolactone, and it's a fairly simple molecule if you compare it to all these other ones, but it's got a couple chiral centers, and then they have these R groups that are attached also. Halichondrins, very diverse and structure compared to the other ones. It has these weird kind of spiroketal linkages between like T ring systems, and a lot of chirality and interesting organic chemical structure. And most of these are all basically natural products. I think this might be an exception based on the difluoride. I don't know that for sure, but these are produced by nature, right? So nature produces some pretty crazy medicines and anti-cancer drugs. Cryptophycins are another kind of macrolactone type thing. What do I mean by macrolactone? It's the cyclic ester, and it's a macrocycle, so that's why I call it a macrolactone. Epoxide, various things. So these bind to microtubules and are anti-cancer drugs. So this is the one, monomethylarstatin, MMAE, that we were talking about. This will be one of the ones we talk about in our two papers that we're getting into. And the slightly better one is tubulysin, core structure. This is a third generation antibody drug conjugate warhead. So this is probably one of the best ones currently used. This was a little bit older, but still interesting also. So we'll call this yellow and this one maybe green. Okay, so for now, notice that MMAE we're going to see MMAE and just recognize that as a second generation one, and tubulysin will be the third generation one. Okay. So tubulysin binds to what's called the vinca domain of tubulin. So this by vinca, they have those alkaloids, which were like I think vinblastine and vincristine, that sort of have this general structure, and I think they're called the vinca alkaloids. And tubulysin binds to the same basically site as those. Tubulin is a polymer, so between beta tubulin and alpha tubulin. So this, as part of kind of how the tubulin separates the chromosomes, it's through kind of a polymerization depolymerization process, and that kind of mechanically separates the two things. So there's tubulysin. It's in its binding site, kind of in between alpha and beta tubulin. It's the same site where vinblastine binds, which is one of the vinca alkaloids. and that was we just showed that one, this crazy molecule here. So this peptide binds to the same place. Here's a question: Why don't we just use vinblastine in an antibody-drug conjugate? If it actually binds to the same place, it probably has a similar effect. Well, this is very complicated to make. This is like a 30-step natural product synthesis. This is kind of peptide-like, and we can make peptides really easy. I could probably make this in maybe 3 weeks or a month if I put my effort into it in large quantities. And these are commercially available in large quantities. Relatively speaking, these are not very difficult to make. That's a very hard thing to make. So yeah, and all those molecules, Taxol and things like that. So that's what's kind of attractive to tubulysin and MMAE, is they're really relatively easy to make, and they're very good at what they do. Okay, so other tubulin binding substances have different binding locations. Just to to show that. This just shows a couple more of these molecules of vinblastine, colchicine, lolamilid, and Taxol. This is inverted relative to the last slide. So it shows alpha, beta, which is different than the last slide. But anyway, vinblastine is another vinca alkaloid. It kind of binds in between at this one binding site between alpha and beta. Colchicine is another of these molecules. A relatively easy structure, simple structure, and it is orange, and it binds to a different place. Lolamilid is a different place, and Taxol is the green one. So it shows that there's lots of different places on tubulin that you can bind molecules and cause this sort of antimitotic effect. All right, so we're getting into the synthesis paper, synthesis of tubulysin. What I'm going to do is just kind of introduce the paper, and then we're going to get into it next time. So you should try to read it, though, if you have time. As Organic Letters, 2009, page 5567. I think it's on the iLearn site, so you can just download it. Total synthesis. What does total synthesis mean? Well, making that molecule, tubulysin, the really good third generation antibody drug conjugate warhead, making it and making it from scratch, right? That's what total synthesis means. So all those other molecules we talked about, Taxol, vinblastine, vincristine, if you Google for total synthesis of Taxol, you'll probably see that the paper I was talking about with my previous advisor. But total synthesis, that concept means make it from scratch, essentially. Okay. And then after we do that, we'll get into this kind of follow-up paper that is really cool. So this is more of a chemistry paper. This is a little bit more of a biology paper on the bottom here. Okay, cool. So so this paper, I'll introduce it now, and then we'll kind of reintroduce it next time and go through it in more detail. First, total synthesis of tubulysin B. So it was an achievement. It was a chemistry achievement to make this molecule by organic synthesis. We love organic synthesis. I love organic synthesis. Maybe you do too. I don't know. So let's talk about the chemistry of this a bit. Here's the abstract, and you can see, well, that's the molecule, tubulysin B, one of the tubulysins. There's a couple of them. First total synthesis of it is described. The aziridine route could not be transferred to the blue moiety. We'll talk about the chemistry of it as we go through, and basically showing the key parts and how they attach them all together. So it's a pretty reasonable chemistry paper. All right, retrosynthetic analysis. What does that mean when you think about a molecule? Retrosynthetic analysis means that they're looking at the molecule and they're figuring out how are we going to make it, but going backwards. That's what retrosynthesis is, right? You should have probably learned about retrosynthesis in your organic chemistry class. So there's tubulysin B, and by retrosynthetic analysis, it's like they looked at that molecule and they're like, how are we going to make this? Maybe find a bond we can make that if we make that bond, it'll break it down into easier pieces. And they decided this squiggly, the little pink squiggly, would be that bond. They thought, well, if we make that, if we make the left piece, make the right piece, we can attach this kind of later in the synthesis. And that looks actually pretty reasonable, because this sort of looks like a tyrosine, tyrosine amino acid, but there's a little bit of weirdness with this extra methyl group and carboxylic acid. So, but that was their strategic decision that they approached this with. So they're like, well, if we do this retrosynthetically, then that'll give us a carboxylic acid piece, right? A carboxylic acid, which is kind of like the left piece, and then a amine piece. And you guys, we and you know how to make amides, right? There's a million ways you can do it, and you've learned throughout this course, maybe EDCI, HBTU, amides are made very simply with peptide coupling. So assuming they make the left piece and assuming they make the right piece, then they could just snap them together with a pretty easy peptide coupling reaction, right? And then they thought, well, we're not done yet. I mean, this thing on the left needs to be made. And so where's going to be the strategic disconnection? Here. And they decided, well, maybe it'll be this squiggly right there. So that was their choice, and thinking that this is probably a pretty easy amino acid type thing that they can attach. And then they can focus on the core structure as a separate task. All right, so let's also go back over here. Basically, they were able to take this, they're making it from kind of a amino alcohol type thing. Bach is a protecting group, you guys know, is a amine protecting group, and essentially their idea is they will, from this building block, which is I think a commercially available or a really easy thing to make, they're able to they'll ultimately oxidize the alcohol to an aldehyde, and then the aldehyde they can turn into an alkene using something called the Wittig reaction. That's an organic 2 reaction that you may or may not remember, and essentially build this kind of bottom piece, with the carboxylic acid. So we'll see that as we go through this. Remember, we're just doing the retrosynthesis now. I'll go through the forward synthesis next time, which is the actual stepping through the forward direction. Right now we're going the backward direction. Okay? But anyway, this is a nice building block that they can start with. Starting from this building block, they're able to basically make this thing and then do the attachment. All right? So then on the left, their strategic approach was that they could attach this thing, this weird N-methyl pyridine amino acid type thing, kind of from this molecule with a weird leaving group, PFP, pentafluorophenol. So this is actually just a key starting material. It's kind of like a chloride, right? This is like just a funny leaving group. But assuming they have this with a funny leaving group on it, then they can couple it. The azide, they can turn it into an amine. It's pretty easy to go from an azide to an amine, NH2. So they'll do that also. So in their real synthesis, they're going to turn the azide into an amine, and then the amine is going to attack this carbon, kick off the leaving group, and then they basically have their key intermediate. Okay. So, Oh yeah, so then it just comes down to how are they going to make this thing? So they're just kind of chopping this up into the fundamental starting materials. Well, this is pretty easy to make actually. They can make that amide linkage through you know, like a peptide coupling, EDCI type thing. So that's a very easy bond to make. And then this is just a kind of attachment of like a protecting group type thing, or whatever. Okay, so ultimately, they begin the synthesis from this material. So they start the synthesis, they're able to attach the bottom thing, and they're able to attach this thing, turn the azide into an amine, amine attacks, kicks off the leaving group, then they got that. So there's basically two fundamental starting materials, this one and this one. And from these two starting materials, they can do the entire synthesis of tubulysin B. Okay, let me see. All right, so I'm gonna just stop here, and I'm gonna go through. I'll go through this next time. I'll probably review the retrosynthesis one more time, and then we'll jump into the actual synthesis of this, and then we'll get into the more of the biology topics. All right, cool. All right, I'll stop now.